So yeah, we are about to start. So before we start, maybe just a logistic issue. If you want to ask questions, I think you can just raise your hand. There's a raise hand button there, and then I'll enable you to speak. Probably leave the questions to the end of the session, so Professor Hall can answer them together. Yeah, if you have a question in the middle of the talk, just actually type down in the chat window. I'll ask at the end of the session to Dr. Huo, so he can answer those questions. Okay, so yeah, it's about time. Let's get started. Good morning, good afternoon, everyone. Thank you for joining the IEEE EMBS webinar series. The webinars are on the frontiers of biomedical imaging and analysis. Today, it is our great pleasure to have Professor Yukai Huo from Vanderbilt University. Dr. Huo is an assistant professor in computer science, computer engineering, and data science at Vanderbilt University, located in Tennessee at the USA. He received his master's degree in computer science from Columbia University in 2014 and a PhD degree in electrical engineering from Vanderbilt University in 2018. Dr. Huo has a very strong track record of working in the healthcare industry with R&D experience. From 2011 to 2014, he worked in Columbia University and the New York State Psychiatric Institute as a staff engineer and research officer. Then he had worked as the research assistant professor at Vanderbilt University, and later a senior research scientist at PAII Labs. Ping An Insurance Company is actually a very good research lab. Since 2020, he has been a faculty member at the Department of Electrical Engineering and Computer Science and the Data Science Institute at Vanderbilt Engineering and University. So his research interests, including medical image computing, knowledge-infused machine learning, and large-scale healthcare data analytics. His research aims to facilitate data-driven healthcare and improve patient outcome through innovations in medical image analysis, as well as multi-model data representation and learning. Today, Dr. Huo will speak about his recent research on scalable learning for large biomedical images. Without further ado, let's welcome our speaker. Thank you.
All right. Thank you, Ping-Kun, for your warm introduction. Hi, I'm Yukai. I'm from Vanderbilt University, and I'm glad to share our recent research in scalable learning for large biomedical image analysis to the IEEE EMBS webinar series. So first, I think most of the audience are engineers or computer scientists. So today, my talk will focus on the technical side of our work with like a little bit clinical background application. So as computer scientists and engineers, so if we want to associate our research with healthcare, I think there are two big directions. So one is bioinformatics. Basically, you can analyze the clinical report, analyze genetic information, blood tests, etc., etc. Another major direction is called biomedical imaging and image analysis. For example, we can use radiology, pathology, microscopy, etc., to understand what's going on inside the human body. So my field is medical image analysis. So this is kind of X to Y elementary school philosophy. Is that what we get is typically X, or some like qualitative images, and we want to associate the image with some clinical outcome, which is Y. And in the middle, we typically have two steps. One is called medical image processing, which is to grab and extract the features and the quantitative informations. And the second step is called data analysis. So basically, after we extract the biomarkers, we can associate the biomarkers with the outcomes. And the entire process we call medical image analysis. So basically, in the medical image analysis, we have several basic tasks. For example, we can understand the entire image called a classification problem or regression problem, or we can try to understand a region in a medical image. So for example, we can do localization, we can do detection. Then more sophisticated task is that we can understand each pixel in the image, which means classification or segmentation. So with the recent technique in GAN, we can do the synthesis. But if we compare our task with computer vision, we see, well, that most of the tasks are kind of one-to-one corresponding to the computer vision. So the next question is, why us? Why do we still need a medical imaging scientist to work on the problem rather than just let the computer vision scientists take over everything? So here is some key points that I summarize. Say we need to solve the unique challenges in biomedical image analysis. For example, we are dealing with high dimensional data. We have strong prior. We have a longitudinal image. We have multimodal data, etc., etc. Okay, so one example is that we are dealing with different signals. For example, in natural image, so for the computer vision, we typically deal with 2D images. But in radiology, we are typically dealing with 3D volume. It is kind of has a much higher dimension than the normal 2D image. Okay, another field is pathological image analysis. So in this case, we're dealing with 2D image, but it's way larger than a normal image in computer vision. For example, this is a kind of quite a large image for computer vision, but if we map it to a whole slide of pathology image, so we can see it's only a very tiny portion. For pathology, we're typically dealing with kind of gigapixel level images. And the last is the biological images, which means microscopy images. So in this case, we are dealing with 4D data because for each time point, we typically acquire 2D or 3D volumes, and we collect data through a fixed number of time points to collect the 4D dynamics for the tissue. And here is the scale. So for computer vision or natural image analysis, it's very easy to scale up to million level images, for example, ImageNet. But here is the typical tasks or questions I got from my collaborators. For example, radiologists typically want to analyze thousands of MRI scans as kind of the maximum number that we have. And the pathologists sometimes say, okay, I have like, for example, 500 whole slide images. So how can you help me to analyze that? For biologists, the scale is even smaller, typically like 20, 30, 50 really high resolution microscopic videos, and how can we analyze the data? So in terms of dimensionality, we have larger and larger dimensionality, but for the size of the data, we have smaller and smaller data. Okay, so another challenge is that the doctors and the biologists, they don't have a lot of time to annotate the data. So here it motivates our research, say, okay, so even we have very small number of limited data in, for example, at Vanderbilt University Medical Center, like this, so this is small scale, but eventually we can get large scale unannotated data, either from our hospital or from public available data set or from our collaborators in another institute. So the outline of my research is try to kind of fully use the smaller scale of annotated data with large scale unannotated data. Okay, so let's see, this is a typical story we have is that we have a set of annotated data. Okay, of course, we can use supervised learning to associate the data with clinical outcome. But the good thing is that now we have a larger scale unannotated data. The first topic of my talk will be the image synthetic learning. Say, well, we want to bridge the gap between annotated data and the unannotated data. In that case, we can still use kind of supervised learning to solve the unannotated data. The second topic is semi-supervised learning. So in that case, so suppose we first have a trained model for annotated data. How can we propagate the information to unannotated data? Third is self-supervised learning. So that is kind of the reverse way, is that we first learn a model from unannotated data, maybe without any specific task. Then we can fine-tune the model with annotated data. Last is called annotation-free learning. Is that possible that we just learn from the unannotated data without annotations? And these tasks are very challenging in computer vision or machine learning community, and especially when we have smaller scale and higher dimensional data in medical image analysis, we typically need to infuse the clinical knowledge and the prior knowledge to our learning framework. I call it kind of knowledge-infused machine learning. Okay, so here is one example. Say we have some kind of unique prior knowledge or clinical knowledge for our data. First, if we can see a natural image and we want to do object detection, which is one of the most standard tasks. So if we rotate the image and use the bounding box as a detection representation, we can see we have very different representation for the same object. And the reason the bounding box is good is that in natural image analysis or computer vision, the scene 1 is common, but we typically don't see the image like scene 2. But in our medical image field, the rotation is quite standard, right? So when we take the picture of a tissue, we cannot control the orientation. And in this case, we propose a new representation called a circle representation for detection. In that case, we can have pretty consistent representation for the same object. So here is more examples. Say we can apply detection algorithm on the same tissue with bounding box representation and the circle representation. If we use bounding box, we can see the inconsistency. And if we use circle representation, the representation are quite consistent with each other. And another advantage is that now we have less degrees of freedom. So we reduce the degrees of freedom to fit detection object from 2 to just 1. So in that case, it will potentially also increase the precision of the detection because the task is simpler. And here is an example that we apply our circle representation to glomerular detection in renal pathology, and it consistently achieved a higher average precision and a stronger rotation inconsistency from this data set. And we also tested this algorithm on nuclear detection, and the same story, we get better AP and stronger rotation consistency. So here examples say, well, if we incorporate the domain knowledge in learning, sometimes we can get like a surprisingly good result. Okay, let's see the first category, image synthetic learning on radiology images. So first example we can see is the cyclogram. So I think that you guys are quite familiar with this image. And we can apply cyclogram to medical image as well. For example, we can synthesize CT from MRI, MRI from CT. But here in healthcare, so the thing we want is not only the synthesis, because there are significant clinical risks that if the physicians directly apply the synthetic image for diagnosis. So in that case, we do one step further. Say our final outcome, we want to have segmentation rather than the synthetic images. So here is the data set we apply this technology. So we do the splenomegaly, which is abnormally large spleen segmentation. So in the case that we have already annotated some MRI scans, and then now we want to apply it to the CT without annotation. And then after using our algorithm, we can get the segmentation on CT without introducing more annotations. So here is the data set. We have unannotated CT, we have MR with annotations, and our goal is to get the CT segmentation without introducing more manual annotations. The idea is that, okay, so here is the standard cyclogram framework. So what we do is that we introduce a new branch called the segmentation head, and we directly apply the segmentation head to the synthesize the CT, and compute the segmentation loss between the estimated segmentation with the ground truth. And now we introduced one loss function to the previous cycle gain framework. And in that case, we can get synthetic result from CT to MR, MR to CT, and we can also get the segmentation on CT. And we also apply this to neuroimaging to see if we can get total intracranial volume segmentation from from MR image without introducing new labels. And here is the quantitative results. And the blue box plot is if we train a CT segmentation model with ground truth, and the red is our proposed framework. And we can see their performance are quite close to each other, and the difference are non-significant. So using the synthetic learning, we can reuse the label and reduce the manual effort. Okay, so here is the second topic, is semi-supervised learning, still on radiology image. So in this case, we show the example with neuroimaging. So the task we're doing is whole brain segmentation to segment the brain to more than 100 regions with less than 50 annotated samples. So why we only have 50 annotated samples is that the brain image, the annotation on brain image is really expensive. It costs $1,000 to $2,000 per brain, and it's very time consuming. Historically, we can use this framework for semi-supervised learning is that we first, for example, using 50 scans to train AI model and propagate the model to maybe 5,000 unannotated images, and we use the manual label and use the label called a Pseudo label to train the second model for our final brain segmentation model. So idea is like this, is that when you have more and more Pseudo label, you can kind of fit the boundary between the classes better and better. And in medical image analysis, here is another useful domain knowledge that we can use, is that we have already developed a lot of kind of solid image quantification algorithm. For example, this is the standard brain segmentation algorithm before deep learning, is that we call it a multi-atlas segmentation. The idea is that when we have an image to segment, we use 40 or 50 annotated example to first register each atlas to the target image to get the deformation field. And then we apply the deformation field to all atlases. And eventually we fuse all segmentation result to the final segmentation and propagate back to the original space of the patient. And now we can get a segmentation algorithm in a totally training-free manner. And if we compare the performance, the multi-segmentation on this task typically achieve 0.75 dice, but the naive unit, so if we train the version 1 of the unit, we typically get 0.6 dice. So that motivates us to not use this strategy of semi-supervised learning. So what we do is that we use multi-atlas segmentation to generate the Pseudo label and train the second model for whole brain segmentation. Except the limited sample size, we have other challenges. For example, the brain images can be very large, and our GPU memory is limited for such large signal. And we only have 45 labels with this large images. So we can do, typically we can do 2D to 3D to 2D, basically design 2D network, or we can do the downsampling, or we can do the random patch. And in our work, we try to solve this problem in a different perspective. So before we introduce our algorithm, we can see, okay, there are many unique advantages in neuroimages that we can use, which is strong prior. So if we compare, for example, we have different horse image, and we have different brain image, like the inter-patient variability between these brain images are smaller than the horse image. So basically, we have strong prior. We have a lot of good tools that can use. So instead of random patch, we first register all the images which to the standard space, for example, the MI space, and we do the fixed location patch. So basically, for every image, we do the patching at fixed location. For example, this is 3D space. Here is one subspace, and here is 8 subspace. Okay, by doing that fixed sample space, we train the corresponding number of different units. For example, we can train 27 different units for different location, and without losing the resolution of each image patch. And finally, we can try different way of separating the fixing space, and eventually we can fuse the result together to even correct some errors from each model. Okay, this is kind of spatial ensemble technology. So basically, we have high resolution 3D data, limited annotations, but we have a lot of good tools. So first, we apply multi-segmentation to get 5,000 pseudo labels, and we train this slant technology with fixed spatial patching locations, and eventually we can get whole brain segmentation. Okay, so here is the result. So we can see the traditional multi-segmentation can take like 36 hours for segmenting each patient. Now we have 5-minute solution with higher dye similarity. So we reduce time-consuming task to 5 minutes. So with this technology, we can do some large-scale clinical study. So we collaborate with clinical neuroscientists to segment 1,500 clinical acquired CT and evaluate performance. So basically, using traditional multi-segmentation, using single machine, we need 2,000 days. And with the deep learning technology, we can segment all 1,500 scans in 5.2 days. With this segmentation, we can do some large-scale data analytics. So for example, we can associate each brain volume with some clinical variables. For example, here we can understand the brain aging to see how your brain volume changes across the lifespan. So we do the task called the brain volumetry. So basically, we can segment the brain, propagate the volume of each region to a plot. And when we have a lot of brain scans, we can have this plot and fit a regression model as the brain development curve. Historically, we do small scale because we don't have a good analytic tool and we don't have large scale data. But nowadays, with a whole bunch of different open source data set, we can easily collect 5,000 MRI T1 images for this task. And with the deep learning technology, we can segment them in an efficient way. And then here is some kind of videos to show how each brain region change across the lifespan. The red means it reached the maximum volume. Blue means it becomes smaller and smaller. We can see how our brain volume changed. And this has been published in human brain mapping to understand the brain development and aging through the lens of inter-network relationship to associate with maybe the socioeconomic status of different subject, etc., etc. Okay, so now I want to introduce the third family called the self-supervised learning. In this case, we use the application is called pathological image analysis. So as we show, the pathological image is very large. Right? So we can see a single whole slide image here for the skin cancer can be larger than the entire CIFAR-10 dataset. And here is the news I saw early this year that the DoD is releasing 55 million of these gigapixel whole slide images in next 5 years. So, but today's technology is not scalable to addressing this scale of whole slide image. The reason is that the manual quantification is extremely time consuming. So it can take hours to just annotate a single whole slide image with kind of different tissue categories. So our motivation is that can we kind of reduce the number of manual annotation through some self-supervised learning strategies. So the technology we use is called contrastive learning. So this is a pretty hot topic in the recent 2 years. the idea is that so we want a network be able to learn from unannotated image to understand that, okay, if we do augmentation on the same images, we still recognize they're from the same dog. And to do that, we typically need to define the positive pairs, basically the augmentation from the same image, or the negative pairs, basically the images from different cameras, different species, or different image from the same species. So then we think, can we use this technology to pathological images as a good unsupervised, self-supervised learning strategy? So for example, we can grab image patches and form them as negative pairs and do the augmentation within each image as positive pairs. Then we realized it would be problematic because the positive and the negative pairs are defined just by how we sample the data from the whole slide tissue. For example, these images we can regard as negative pair since they are different images. But if we sample the image in the yellow box, these become positive pair because they are from the same image. Okay, so then the next question is that can we use the technology to only have positive pairs? So fortunately, later in 2020, we have these good technology, WiseBio and the CMCM, to deal with the contrast learning with only positive pairs. But they either use a momentum encoder or just ignore the momentum encoder. So here is the new technology we propose called the SIM triplet. So we got this idea from triplet loss, triplet learning, that we introduce another branch from the image. So because we can sample the nearby patches from a whole slide image, and the additional patch can provide the momentum explicitly without the momentum encoder. So by doing this, we have momentum, but without extra memory consumption. So we have extra momentum, and it can be run on single GPU. We use the multi-view nature of pathological image, and we don't use any positive pairs. And similar to triplet loss, now we can also minimize the intersample, and we can maximize the intersample similarity between the image samples. And here's an example. So if we only have small number of annotations, the supervised learning basically crashed. But with the self-supervised learning using SIM triplet, even 1% level can give us really good segmentation performance. And since we have the explicit momentum, the performance is better than CMCM. And now we are doing some downstream analysis that use the features extracted from self-supervised learning to the survival analysis. We can see our SIM triplet can get significant survival result compared with bio and the CMCM. Okay, so we introduced the self-supervised learning on cancer pathology, but how about another family is called the non-cancer pathology? So in that case, we are interested in the isolated objects on a whole slide image. For example, this is renal pathology, and if we're interested in glomeruli, we first need to identify all glomeruli rather than just sample the nearby patches. Then, once we have each glomeruli, we can again apply contrast learning for some downstream tasks. But the challenge is where can we get the data? Because for each individual laboratory in university, typically we limit ourselves to like 100 or 200 whole slide images for kidney, et cetera. So our idea is to grab data from online resources, whether from Google, from NIH PubMed, from open access journals. Okay, so then we find the problem is that if we download the data automatically from this data set, we can see in academic publication, we typically put the data as subplot in a bigger picture. So we basically need a technology to detect all each components and be able to tell us, for example, the yellow parts are the images that we need. Okay, so the task is called compound image separation. So instead of annotating the images as training data like the right side, So we use the technology called Pseudo training. Basically, we first collect some single images online and assemble the images to some fake annotated detection annotated images. And to further improve the performance, we introduced the intra-class augmentation, basically to put the same classes together as detection training data. And beyond that, we introduced a side loss for our unique task. The reason we introduced that is that the traditional IOU loss will prefer to detect a larger box rather than a small box. So that's why we introduced the side loss to encourage the network to detect the inner part. So here's the visualization. The red is the traditional IOU loss, and the yellow is our side loss. By using all these different technologies, here is the result that we can get from our algorithm without using any annotation result for detection task. And we achieved even better performance than the state-of-the-art performance on the famous image clip 2016 dataset. Okay, so from this technology, what we want is to get huge number of unannotated data. So we collect 20,000 unannotated glomerular image from PubMed using this image separation technology. And then we can apply this data set to some downstream tasks. Again, we do the self-supervised learning, contrastive learning to pre-train the model and apply to different tasks. First is the stand classification task. It's just like in radiology, we want to curate different contrasts for CT, et cetera, et cetera. So now we can classify different stands to different categories. And we achieved better results than supervised learning. And here is another downstream test, which is more interesting for physicians, is that we can now do the fine-grained glomerular lesion classification with a small number of training data. For example, we achieved a better performance than supervised learning on this 5-class classification. So this work is still under review. Okay, with this 5-class classification, now we can have this detection and the quantification result on a whole slide image. Now we can see different colors means different classes, and we can get the lesion type of each glomerular. And then what we want is that, okay, so here is the 2D result, but if we have different cuts of the same kidney tissue, for example, if we have like a 17 or 16 sequential cut, can we do the 3D analysis like we do in radiology? Then we propose a new technology to associate different 2D slides into a 3D association. We call it Map3D. And this technology is totally training free, which means we don't use any annotated data in training. We use super glue model to do the global association and use traditional retrition in neuroimaging to align the image at fine scale. So by doing that, now we can align each glomerular in 3D space. For example, in the light blue box, we can see here are all cuts of the same glomerular. Then we can quantify the glomerular in 3D. And our Map3D algorithm achieved better result than previous 3D pathology retrition method. And with that, we can do some downstream tasks, for example, to understand this specific lesion called atubular glomerular. And traditionally, it takes 30 hours for a single mouse kidney for human. And now we reduce the time 60 times to half an hour. Okay, so now I will introduce the last piece is called annotation-free learning. So in this case, so what we want is to do the learning using the unannotated itself. So the application here is biological images, which is microscopy image. So the task we are dealing with is instant cell segmentation and tracking. So here example, say we want to segment each cell and track the trajectory of each cell. This problem is quite similar to the multi-object tracking in computer vision. You can see in MOT, we're also trying to identify each object and track their trajectory. But there are many unique challenges in biomedical image analysis. First, we have mitosis, which we frequently see a cell split to two cells, which is not quite normal in computer vision. We don't see a human split to two humans. And the data, again, we have very small scale of data, but in very heterogeneous domains. Here are some data from the famous cell tracking challenge. So since the data are heterogeneous, we have a lot of mitosis, and the sample scale is small. So here is how traditionally biologists are dealing with these data set. Is the traditional model-based image segmentation. For example, they can use cell profile or image J to design the workflow and to segment and track the cells. Okay, and then we say, okay, can we develop some deep learning technology for this task to replace the time-consuming manual design efforts? Okay, the interesting is that if we see image classification in medical image analysis, no matter in radiology or pathology, well, we have some standard tool can use from computer vision. For example, we use ResNet for classification, UNet for segmentation, mass Gaussian for instance segmentation. So this has been widely accepted. So basically for a new task, if we don't want to have new development in terms of technology, we can directly use these existing tools. But for instance segmentation and tracking, we still don't have like this type of easy to use end-to-end tool. And until 2019, we saw a great idea from Payer's group is that they defined the instance segmentation using the domain knowledge in biology. Traditionally in computer vision, for example, we have like 11 cells to segment. So basically we want to have different features or different representation for all these 11 cells, okay, if we use traditional computer vision. But Payer has an awesome idea, say, since the things are kind of duplicated in biology or in microscope, we don't necessarily need to have 11 different representation for 11 cells. We only need to make sure the representation is different across the neighboring cells. So for example, in the middle, if we see a blue cell, we only need to have different representation from the nearby cells to that blue cell. We don't necessarily need to care about other cells. And we don't even need to have different representation between the cell number 4, number 3, number 11, and number 10. So this is a transformative idea to do the instance segmentation and the tracking in biology. And that's the reason this paper won the Yukai 2019 Best Paper Award candidate. Okay, and using this idea, we can also solve the tracking problem simultaneously in the same network in the end-to-end training. And we are very excited, and we apply this method. But the thing we find is that the method is quite slow because this is kind of representation learning based method. You need to kind of cluster the features after you learn the representation. So then we propose a new fast algorithm using GPU acceleration. We call it fast mean shift to reduce the processing time from basically half minute per frame to like 2 to 3 seconds per frame. So here is the detailed technology. So the basic idea is that we, from the same hypothesis that the biological image or microscopy image has the target in a repetitive way, so we can compute the lower bound of the seeds that we need, which dramatically reduce the time of the clustering process. And here is some result that our method achieve the fast clustering and with the minimal GPU memory consumption. Then we realized, well, the Yukai 2019 method can only deal with 2D images. But in our reality, the advanced microscopy imaging are collecting 3D data like the right side. So we can scan the image in 3D using the confocal light sheet microscopy or light sheet confocal microscopy or light sheet microscopy. So we need a technology to kind of change the way of analyzing the instance segmentation and the tracking from 2D to 3D in the end-to-end manner. So here is our idea. So our idea is that, okay, so since the 3D is time consuming, we don't want to use the 3D convolution kernels like the standard operation. What we want is to redefine the problem as representation learning. So here is the idea of the 2019 Yukai work, is that how they associate different time point is to force the same cell from different time point with the same representation. So we say, okay, this technology can be easily extended to space as well. So we can regard the different space, the slice from different space as a time, right? So you can, if we go through the 3D, you can see the cell changes. So in 3D, it's kind of become larger, then smaller, and eventually disappear. So it's a similar thing as a time. So we think we can integrate these two dimensions into a holistic, unique framework. And the idea we propose is called Zigzag Training. So for example, if we have 3D cells, so here is the 3D cells. This is a particular slice across the time point. And then we can have different slices in 3D. So one dimension is 3D, another dimension is time. So instead of only train the blue lines, we introduce these paths as well to cover all 2D slices in this spatial temporal space. And by doing this type of training, we associate the 3D information just using a 2D network. And for each pixel, we have extra 3D embedding beyond the traditional 2D embedding. So we call it voxel embedding. So by doing this technology, we achieve the 3D cell instance segmentation and tracking using a 2D network in the end-to-end training framework. And this network can be done using a single GPU without introduce extra significant memory consumption. So here is an example from the cell tracking challenge. You can see we can get pretty decent 3D instance segmentation and tracking result. And the interesting thing is that even with limited manual annotations, for example, we have like a sparse slice annotate, which is typical in 3D scenario because it's very time consuming to annotate each frame in 3D and in time domain. So if we only have sparse annotation, our method can also achieve really good result because it is a very flexible framework in that zigzag training strategy. Okay, now we can use the sparse annotation. So next question is that can we even go one step further? Say, can we do purely like an unannotated way to get the instance cell segmentation and tracking result, which avoid the manual annotation? The motivation from our collaborators say, so he want to segment and track all these microvilli. Microvilli is a very important subcellular structure related to our nutrition absorption. So our idea is say, okay, so can we disentangle the cellular image to 3 different parts: shape, appearance, and the dynamics. The shape means the mask, basically. So if we have some prior knowledge to say what the shape the object would look like, for example, for the microvilli, we can suppose the shape like a stick. For cell images, we can suppose the shape were like a ball or a circle. And appearance, we have kind of different, we can use again to mimic the appearance of the cell. And for dynamics, we can kind of try to model the temporal dynamic, which is motion of each object. So the idea is that if we can synthesize the image from the stick to the microvilli, we have the hope to mimic different scenarios and extend the synthesis from just the image level to the video level, and to get a whole bunch of training examples for free. Right? Considering if you have these images, videos without their ground truth annotation, you can use the Yukai 2019 method or our GPU accelerated method or even the 3D voxel embedding to train the network using this dataset. then the entire pipeline will be totally annotation free. Then the key thing is that how can we do this synthesis? So instead of doing the synthesis from the instance annotation we simulate, we use the grayscale image in the middle. Okay, so we try to do the synthesis between the middle column and the rightmost column in this framework. So we have the simulated ground truth, we simulated the grayscale image, and we build the image synthesis network using CycleGAN. Then we can get a whole bunch of different synthesis image from these binary masks, and eventually we know the ground truth of these binary masks. Okay, so then we have the training pair, say the intensity image and the instance. The good thing here is that there are many like overlapping or kind of mitosis dynamic situation, which is typically really difficult to annotate. But since we do it in the simulation way, we can simulate all these different situations. And we also extend this idea to cell segmentation, and we can see we can synthesize pretty good cell segmentation result. And all these synthesis cell segmentation training data from the ball shape annotations. So the details you can refer to our paper. So basically, using this strategy, we can get large-scale training data for free and significantly boost the performance. Okay, so here are basically all different topics I would like to introduce today. And then the question is, why do we want to understand so many different modalities using different scalable learning algorithm, right? So why do we need to do radiology, pathology, or microscopy images? So here is the answer. So here is the project launched in 2017 called the Human Cell Atlas. This is kind of global effort to try to understand the human tissue and the human cells. And the Vanderbilt is mostly contributed to the part called the Gut Cell Atlas. So the Gut Cell Atlas is trying to understand the IBD disease, Cohen's disease, and try to understand the cells in our human gut system. And the right side are the data we have from Vanderbilt. So we have H&E images, which is the microscopic image. We have MXIF, a lot of, it's also microscope and also pathological images. And we have radiological images, sequencing, and we have clinical information at the end. So I think this is the trend, is that so to understand the disease, understand the clinical problem in a more comprehensive way, we need to understand the different modalities and be able to integrate different modalities together. And hopefully our solution will be the way to kind of solve this complicated cell gut atlas problem. And if you're interested in this problem, and you can shoot me email, and I'm happy to collaborate with you guys to tackle this complicated data set. Okay, that's all my presentation, and thanks for, I'm fortunate to work with these smart guys in our lab, and thanks for all these funding agencies to make our work happen. And thank you very much.
Thank you very much, Yukai, for the excellent talk. You cover a lot of intermediate methods. That's great work. Now we can actually open the floor to the audience for questions. So if you have any questions, you can either type in the Q&A window or just raise your hand. I'll unmute you. You can ask questions. Either way, it works. Okay, maybe I can start asking the questions so the audience can prepare their own questions. So I see actually the first point you talk about using the circle representation in the pathological image analysis. I think it's a very interesting idea. But when you cut actually circular representation, how do you fit into the CNN? Because CNN typically take the the rigid, the grid structure data.
Yeah, that's a great question. So we use the backbone is from the CenterNet. So for object detection, so the first thing is to fit a heat map, which shows, okay, here is the object, here is the elephant, here is a cat, etc., with a heat map. Then the traditional way to fit the bounding box is to fit two-dimensional feature of each hotspot. So basically, if you have 100 by 100 image, now you have 100 by 100 by 2 feature map to show if there is a bounding box, what is the height and the width from the center point. And by doing that, we reduce the feature map from 100 by 100 by 2 to just 100 by 100 by 1, because now we only need to fit the radius. We don't need to fit the height and the width. So in that case, if we know the center point, if we know the radius, then we can draw a circle. So we not directly fit the boundary of circle. What we do is that we fit the radius.
But the data representation in the typical CNN, we just got a 2D array in some sense, actually a tensor, right, into the neural network.
Yes.
Because you have the circular shape, I mean, actually inside the pixel, they are not actually rectangular shape. How do you handle that?
Yes, you're right. So basically, the shape of each pixel is kind of assembled in rectangular way. But one way is that to fit the mask-like stuff. If we want to segment the tissue with the circle mask, that would be problematic, because now you need to deal with, okay, how to split this pixel at a circle boundary, or how can we fit a convolution with this circle, etc. But now we fix one value for each location. So in that case, the CN, I think, it try to understand, okay, so now I see object. I just want to estimate how big it is. The CN actually learn that information rather than explicitly draw a boundary or the mask. They just try to learn, okay, here has an elephant and how big the elephant is. Then once we have this variable, we can draw a circle. So this is kind of a different perspective of this kind of circle representation. So we don't explicitly learn a circular mask. We just learn the radius.
Oh, okay, I see. Yeah, that's a very smart idea. Yeah, very interesting. Thank you. So in general, I can see actually you list several challenges. Indeed, actually, people working in this domain, medical image analysis, medical image computing, face those challenges, right? Limited data sets, limited annotations. You have been applying the different kinds of methods trying to mitigate such challenges. So from your point of view, what do you think actually the next step in this field, right? We cannot always borrow the techniques from computer vision, machine learning, like semi-supervised learning, self-supervised learning, etc., etc. So what do you think maybe actually next breakthrough?
Yes, that's a really great question. So as far as based on my collaboration with clinicians, now they are trying to really interested in the idea called multi-view learning. So basically, traditionally we focus on one modality. For example, if we want to understand the fatty liver, we focus on liver. But then the physician's idea say, well, if you can solve each modality, how about if you can fuse all different modality together? Because in the clinical practice, the physicians need to consider overall information from a patient then to make a diagnosis. So currently our ongoing work is to first be able to extract reasonable feature from each modality, like shown in cell gut atlas. Then the important thing is that how to fuse these information together to the final clinical outcome or explore new biomarkers, etc., etc. By doing this fusion, we also can improve the feature extraction. So they will be integrate end-to-end system eventually to help us to learn the disease or solve the clinical problem better. And this is kind of our ongoing project.
Yeah, great. Yeah, I completely agree. It's very important for us to not only understand the technology, but also actually go deep into the applications, right, to find out the specific challenges in the problems, trying to solve them.
Yes.
Great, thank you. Yeah,
Thank you.
I was also very glad to see actually you're going to present your paper at machine learning in medical imaging workshop. The floor was great.
Yes.
All right.
Yes.
Okay. Any questions from the audience? So there was one question. Let me see. Can do feature selection in network. Can you please actually elaborate your question? I'm not sure. Maybe Yukai, you know what's the question about.
Okay, so the feature selection. Okay, so the feature selection. I think the problem is that how to do the feature selection to associate the large signal to final clinical outcome. For example, for radiology, we have a huge 3D, but if we only have the diagnosis, say this guy is Alzheimer or not, if we have huge pathological image, but we only have the diagnosis, say if this is a cancer or non-cancer, how can we extract the useful features from the huge feature space? This is my understanding of the question. Correct me if I'm wrong. So if the question is like this, so my idea is that we also need to incorporate the beautiful clinical knowledge beyond the traditional multi-instance learning or those kind of feature extraction strategy. For example, for liver cancer, we can focus on the tumor region rather than look at the entire CT volume for the cancer diagnosis. So that will dramatically reduce the feature space. And for like the renal pathology, we can limit ourselves to glomerular or even the tissue in glomerular from the previous clinical knowledge to reduce the feature space that we need to learn. So this is kind of my understanding of how to reduce the feature space.
Okay, thanks. Yeah, I think you answered the question, so you typed it. Thank you from Yuzhou. Okay, great. Any other questions before we conclude the session? Okay, if not, let's thank the speaker again. Thank you, Yuankai, for the excellent talk. Thank you very much.
Thank you, Pinkun, for organizing the excellent series. Thank you so much.
Okay, thank you everyone for attending the webinar. I'll see you next time. Bye.
Bye. See you.